Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the final decade of the 20th century, biology confronted a challenge of breathtaking scale, a task whose ambition was commensurate with the very code of life it sought to decipher. The objective was the complete sequencing of the human genome, a molecular tapestry comprising 3 billion base pairs. The international, publicly funded Human Genome Project began this monumental journey with a strategy of meticulous, almost scholastic, precision. Its chosen methodology was a hierarchical, clone-by-clone approach. This process involved first breaking the entire genome into large, manageable fragments of roughly 150,000 base pairs and cloning them into bacterial artificial chromosomes, or BAC. These BACs were then painstakingly mapped, assigning each large fragment to its specific location on a chromosome, creating a comprehensive physical map. Only after this cartographic foundation was laid would each individual BA cell be selected and its DNA fully sequenced. This was a plan of immense intellectual integrity, promising an authoritative, finished reference sequence. It also promised a timeline stretching well over a decade, a pace that felt glacial to some observers eager for more immediate biological and medical insight. Into this environment of deliberate, methodical progress stepped Craig Venter, a researcher at the National Institutes of Health with a different sense of urgency. He surveyed the grand architecture of the public project and perceived a fundamental bottleneck: the most valuable territories of the genomic landscape, the genes themselves, which constitute a mere fraction of the total DNA, would be among the last regions to be fully characterized. Venter proposed a radical departure, a strategy that prioritized rapid gene discovery over comprehensive genomic mapping. His first major contribution, formulated in 1991, was the Expressed Sequence Tag, or ESD, a method that was both a technical shortcut and a profound conceptual reorientation for the field. It shifted the primary goal from sequencing an entire encyclopedia to first compiling its index of key terms. The elegance of the ESD strategy resided in its direct exploitation of fundamental molecular biology. The process began by isolating messenger RNA, or mRNA, from cells. Since mRNA is transcribed only from active genes, this step immediately filtered the vast expanse of the genome down to the functionally relevant protein coding sequences expressed in a given tissue. Venter's team, particularly focusing on the human brain for its rich diversity of expressed genes, then employed the enzyme reverse transcriptase. This enzyme transcribed the single-stranded mRNA molecules back into more stable, double-stranded DNA copies, known as complementary DNA, or cDNA. These DNA libraries represented a snapshot of the active genetic profile of the source tissue. The crucial innovation lay in what happened next. Instead of undertaking the laborious task of sequencing each cDNA molecule completely, Venter's approach performed a single, automated sequencing run from one end of each clone. This generated a short fragment of high-quality sequence, typically a few hundred base pairs long. This fragment was the Expressed Sequence Tag. An ESD was an imperfect piece of data. It was a short, unmapped fragment from an unknown gene. Yet, its utility was immense. Each ESD served as a unique identifier, a flag in the ground announcing the existence and expression of a particular gene. By generating hundreds of thousands of these tags, Venter could rapidly create a vast catalogue of a significant portion of all human genes, long before the public project could finish sequencing a single chromosome. This technique leveraged the foundational deoxysequencing chemistry developed by Frederick Sanger, but applied it with an industrial philosophy of high-throughput automation. The direct impact was revolutionary and almost instantaneous. Data, comprising thousands upon thousands of novel human gene fragments, were deposited into a public database, GenBank's DBESD. For molecular biologists around the world, this was a watershed moment. A researcher studying a specific protein could now search this database, find a matching ESD, and immediately obtain a DNA clone for that gene. This process bypassed years of tedious, resource-intensive work that previously involved screening genomic libraries. Venter's ESD strategy furnished the global scientific community with a powerful new tool for discovery, fundamentally accelerating the pace of biomedical research throughout the 1990s. It was a pragmatic and powerful demonstration that in the quest to understand the genome, obtaining useful information quickly could be profoundly more impactful than the slow pursuit of ultimate perfection. This initial foray set the stage for a far more audacious proposal that would challenge the very foundations of the public genome effort. Craig Venter's ambition, however, dwarfed even the ISD project. He proposed a radical acceleration of genomics sequencing, the whole-genome shotgun approach, or WGS. This strategy, coupled with his founding of Celera Genomics, ignited a firestorm within the scientific community and fundamentally reshaped the Human Genome Project. The Human Genome Project initially adopted a meticulous, stepwise approach known as BAC sequencing. This involved breaking the genome into large, manageable chunks, cloning these fragments into bacterial artificial chromosomes BACs, carefully mapping their positions relative to one another, and then sequencing each BAC individually. It was methodical, reliable, inventor. Agonizingly, WGS, by contrast, embraced a seemingly chaotic approach. The entire genome was sheared into millions of random fragments, typically a few hundred to a few thousand base pairs in length. These fragments were then sequenced en masse, without any prior knowledge of their location or order within the genome. The true challenge lay in computationally reassembling these disparate fragments, like reconstructing a jigsaw puzzle where the pieces were duplicated, distorted, and lacked any identifying markings. The key to this reassembly was the presence of overlapping sequences. By identifying regions where fragments shared identical or highly similar sequences, computer algorithms could piece together the larger contiguous sequences, or contigs, and ultimately reconstruct the entire genome. Venter staked his reputation and considerable private capital on the claim that WGS could achieve what the public consortium could accurately sequence the entire human genome faster and at a lower cost. This challenged scientific orthodoxy and provoked skepticism from many HEP leaders, who saw the shotgun approach as inherently imprecise and prone to errors, particularly in repetitive regions of the genome. The publication of draft human genomes in February 2001 in Science, Celera, and Nature HEP served as a dramatic validation of WGS's viability. While both drafts contained gaps and errors, they demonstrated that the shotgun approach could, in fact, generate a reasonably complete and useful genomic sequence. The Celera paper, led by Venter, explicitly showcased the power of their assembly algorithms, detailing the computational strategies employed to overcome the inherent challenges of WGS. This included the use of paired sequencing, where the sequences from both ends of a DNA fragment could be determined, providing crucial long-range information for anchoring contigs and resolving ambiguities. Specifically, they applied advanced clustering algorithms, akin to those used in data mining, to identify the most probable overlaps between fragments, even in the presence of sequencing errors or repetitive sequences. The algorithmic details were not merely a technical footnote. They were the intellectual core of Celera's approach, and the primary justification for their claims of superior speed and efficiency. The success of WGS directly impacted the public consortium, forcing them to accelerate their timeline and incorporate elements of the shotgun strategy into their own work. The HEP adopted the use of larger-scale sequencing centers and improved computational methods for assembly, directly stimulated by Celera's advances. It became clear that a purely BAC approach would prove economically and temporally unsustainable. This change in strategy demonstrated the power of competition and how even a scientifically controversial approach could drive innovation across an entire field. The influence of WGS extends far beyond the initial Human Genome Project. Its dominance in modern genomics is almost absolute. The 1000 Genomes Project, which aimed to catalogue human genetic variation, relied heavily on WGS to analyze the genomes of thousands of individuals. Similarly, the field of metagenomics, which studies the genetic material recovered directly from environmental samples, utilizes WGS to characterize complex microbial communities. The ability to rapidly sequence and assemble genomes from diverse sources has revolutionized our understanding of biological diversity, evolution. Even in the specialized area of ancient genomics, WGS played a transformative role. Svante Pääbo, a pioneer in this field, used WGS to reconstruct the genomes of Neanderthals and other extinct hominins. The fragmented and degraded nature of ancient DNA presented significant challenges, but the power of WGS, coupled with sophisticated computational methods, enabled Pääbo and his team to extract invaluable genetic information from these ancient remains. Critiques did certainly emerge. Some scientists were concerned with the proprietary nature of Celera's data and the potential for limiting access to genomic information. Others questioned the accuracy of the WGS assembly, particularly in regions of high complexity or sequence repeats. However, these debates catalyzed improvements in sequencing technologies, assembly algorithms, and data sharing practices. The competition spurred the development of open-source bioinformatics tools and contributed to a more collaborative and transparent research environment. The Venter-HEP rivalry transcends a mere race to sequence the human genome. It represents a productive dialectic between public, methodical science and private, Disruptive innovation, Venter's audacious gamble with WGS challenged established norms, accelerated the pace of discovery, and ultimately transformed the landscape of genomics. The enduring legacy is the global adoption of WGS as the standard methodology and the fundamental alteration of scale, speed, and economic model of big biology. The controversy that surrounded Venter's approach underscores the tension between open access and intellectual property, but it also highlights the crucial role of competition in driving scientific progress.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.